record. So thank you all for joining us for what I definitely think is going to be a great talk. And thank you for Jonathan for agreeing to talk to us today. Jonathan's the Bing Professor for Population Studies in the Departments of Genetics and Biology at Stanford University. He received his BSc in Biology and Mathematics from Penn State University in 1994, and then did his PhD in Biology at Stanford in 1998. He moved to Oxford in the Department of Statistics for his first postdoc, and then got his faculty position at the University of Chicago in 2001. From 2008 to 2019, he was an investigator of the Howard Hughes Medical Institute, and in 2013, he'd returned to Stanford for a faculty position there. Throughout his career, he's received various honours and awards, including the Fabio Frazzetto International Prize in Physical Anthropology from the Lincean Academy of Italy, the Kistler Prize in Population Genetics and Society. He's a fellow of the American Academy of Arts and Sciences, and he received the Edward Nowitzki Prize from the Genetic Society of America. Jonathan's work has made important advances in the use of statistical and computational methods to questions in genomics and evolutionary biology. His research spanned a variety of organisms and has answered questions about evolution, health, and history. Many of the methods he's developed have quickly become standards in the field and are used worldwide. His recent work has been understanding the genetic architecture of complex traits, and it's this work that he will talk about to us today. So thank you very much, Jonathan.
All right, great. Thanks, David. Thank you so much for the invitation to be here, and really looking forward to talking with all of you. If you have any questions as we're going through, then you can just jump on the Zoom and ask, or if you like, you can put them into the chat, and David's promised to monitor the chat and ask. And I think having some interaction during this is probably a nice thing. So. Okay, I will share my screen. Okay. All right. So can you see, are you seeing the presenter mode, or are you seeing
we're seeing presenter view.
Oh, okay, one sec. Let me try. How's this working?
That looks good.
Okay, great. All right. Okay, so today I'm going to tell you about work that we've been thinking about in my lab in the last few years, in which we're trying to answer this basic question of why are human complex traits so extraordinarily polygenic. And I'll tell you in a minute what I mean by that. I should acknowledge the people who've been involved in this before I go on. So this is a project that was started in some senses by Evan Boyle and Yang Li when they were in the lab, and continued by Chuanyao Liu. And most recently, NASA, Sinat Armstrong and Sahi Naqvi, who are in the lab now, have been doing work on the genetic architecture of some biomarkers, molecular traits that I'll talk about in detail. So I think it's fair to say that historically a lot of our intuition in genetics, particularly in the 20th century, came from studies of simple or single gene traits. So, of course, if you think back to Mendel's work, Mendel's great fortune, I think, was that he picked some traits that just happened to be simple monogenic traits, and that allowed him to derive the insights that he did. And then, of course, in human genetics, if you think about the history of gene mapping, in the 80s and 90s, there was an enormous amount of success in identifying genetic variants and mutations that underlie monogenic disorders. In parallel, people were trying to identify genetic variation that underlies complex traits, and that, of course, proved at that time to be much, much more difficult. And I think it's fair to say that a lot of the ways that we think about genetics traditionally comes from these kinds of these kinds of processes where you have a mutation knocks out a gene, and that that leads directly to a phenotype. But of course, we now know that most phenotypic variation is genetically complex. So height, for example, here, and what I'm going to argue to you is that the mapping of genetic variation for a trait like height to phenotype is both much more complicated, but also different than what we see for monogenic traits. And so the fundamental question that I want to ask is, why is it that so much of the genome affects any given complex trait? So I'm going to start out by giving you some examples. First from the literature. Of course, there's an enormous number that I could have shown you. I'll just show you some illustrative cases. So the first is for schizophrenia. So the latest published meta-analysis for schizophrenia incorporated about 150,000 individuals. They identified 108 genome-wide significant loci. So that's an extraordinary accomplishment. But it was pointed out that actually the hits themselves are only responsible for about 10% of the expected heritability for schizophrenia. At the same time, it turns out that using methods that can estimate the total SNP-based heritability, the genetic variation does explain about 80% of the total heritability. And what that means is that there's actually an enormous number of additional variants that are lurking down here in the bottom of the Manhattan plot that do have effects on schizophrenia risk, but in the current sample sizes, they are not genome-wide significant. So the variants that creep above this genome-wide significance line, although there's more than 100 in this meta-analysis, these are really the tip of the iceberg. There's an enormous number of variants of small effects that together are responsible for the bulk of the risk. The last point I want to make on this is that for schizophrenia, as well as for most other complex diseases, a lot of the lead hits are difficult to interpret. A few of these hits, particularly this chromosome 6 hit, have been dissected through enormous amounts of molecular work, but in general, in most cases for complex traits, they're difficult to interpret. In this study, they used They used various gene ontology and pathway kinds of analyses. And in fact, they found that none of these gene sets were actually significantly enriched, although I should note that the top scoring gene sets in this study were involved in processes like synapse formation. And so there's a hint that actually what they're getting at the top of the list for the pathways are probably real, even though the signals are weak enough that they're not actually reaching genome-wide significance. And I would say that these kinds of patterns are typical of a lot of complex diseases that have been studied in great detail. The next point that I want to make is that actually most of the genome contributes to schizophrenia's heritability. So this is a plot from Bogdan Pasaniuk's lab that was published a few years ago, looking at the same schizophrenia data. And what they're doing here is that they're taking each of the chromosomes and they're estimating how much of the heritability is explained by each chromosome. And so each of the blue bars here is showing you one chromosome. The x-axis is using the number of SNPs as kind of a proxy for size. The y-axis is showing how much heritability is coming from this chromosome. And you can see that actually basically the amount, you can predict how much heritability is coming from each chromosome simply by its size. There's a small outlier, which is chromosome 6 here. That's the chromosome where that single biggest peak was on the Manhattan plot. So that's what you might expect in a scenario where you have heritability spread quite broadly across the genome. I'll just show you a contrast to rheumatoid arthritis, in which the MHC region makes an enormous contribution. So MHC on its own is responsible for about 20% of the SNP heritability. At the same time, if you look at all of the other chromosomes, you can see there's a polygenic background that's contributing quite a lot of the heritability as well. I think it's fair to say that actually most complex traits look more like this than they look like the RA example. There's been a number of studies, including from our lab, that have tried to estimate how many variants across the genome affect these traits, and we've estimated that the number may be as high as 100,000, and other estimates from other groups are in the same kind of range of definitely more than 10,000 and up to around 100,000 SNPs, spread, with very small effects generally and spread very broadly across the genome. So I think this raises a question. If it's the case that you don't have any individual variants that make major impacts on schizophrenia risk, the lead variants are not especially enriched in functional pathways that you would a priori expect to be involved, what's the nature of this mapping from genetic variation to phenotype? And so to make some progress on this, in the next part of the talk, I'm going to explore in a bit more detail the architecture of two traits that we think of as kind of being model traits, urate and testosterone. And we got interested in these traits because they sit somewhere in terms of complexity, they're much more simple than traits like schizophrenia and height or diabetes or other disease traits that have been looked at. We think that we can actually, from the GWAS data, we find that we can actually understand a lot more of what's going on. And so we think that we can use these as a system to learn some general principles of this mapping from genetic variation to phenotype. And in the second half of the talk, what I'm going to do is to propose a conceptual model for this mapping. And one point that I want to make is that we think that cis and trans gene regulation is really a key component to understanding the architecture of complex traits. Okay, so the first example I showed you was published work from schizophrenia. I now want to talk about urate levels. So urate is a small molecule that circulates in the blood. It's the levels of urate in the blood are tightly regulated. If you have too much urate, this causes gout. And gout is basically this process where the urate crystallizes into these sharp needles in the joints, and it's apparently extremely painful. I should say that this work's been led by two wonderful people in my lab, Nas and Sahin. So one reason why we became interested in urate is that there's very good data on this from the UK Biobank project. So UK Biobank measured urate as well as about 30 other small molecules in urine and plasma and serum from about 430,000 individuals. For urate, we're going to be looking at the serum data. And so one point I want to make is that unlike schizophrenia, where we don't really know a lot in advance about the processes that control schizophrenia, for urate, there's actually a lot that's known in advance. And so one thing that we know in advance is the full molecular pathway that produces urate levels, that produces urate. So urate is basically a byproduct from this purine synthesis pathway. Everywhere there's an arrow on here, we actually know what the genes are that are catalyzing that step of the metabolic process. And so I'll get back to the individual genes in a minute. Secondly, we know a lot about the transport processes that are controlling urate in the bloodstream. So urate is basically pulled out of the blood into the kidneys by these transporter channel genes, and so basically you've got this export of urate from the blood into the urine. And then once it's inside the kidneys, it can either be excreted out, or it can be reuptaken into the bloodstream to maintain appropriate levels of urate. And so all of these channel genes are genes whose identity we know. And so this is a situation that's very different from something like schizophrenia, where a priori, before you look at GWAS, we really have very little idea of what the relevant genes should and even processes should be. Okay, so here's the Manhattan plot for urate levels. And first point I want to make is that even though it might seem a priori like a relatively simple trait, we get more than 200 independent genome-wide significance signals. The second point I want to make is that most of the lead hits are immediately interpretable based on the known biology of urate. So the peaks that are labeled in green all contain one of those solute carrier genes that I mentioned before that are annotated from known literature. Additionally there's a group of hits that are marked here in blue that are all important transcription factors that are involved in liver and kidney development. And then in red there are a couple of other hits that actually we don't understand very well, but I would note that these same hits are actually involved in are also hits in a lot of the other biomarkers that we've looked at. So they're doing something important probably in overall kidney function, even though we don't really understand what that is. So one point I want to make here is that actually the situation is really different from what you get when you look at like a Manhattan plot for schizophrenia, where putting these, it's extremely difficult to do a small amount of work and label each of the peaks based on prior knowledge.
So John, can I just question on the level of complexity already here? So this is a GWAS analysis of an ascertained population with a problem of urate metabolism. So if you were just to look at normal variation of
this is from UK Biobank. This is random individuals.
Random individuals. Okay, thanks.
Yes.
Okay.
Yeah. Sorry for that. Yeah. So it's individuals in UK Biobank, and essentially everybody who's in UK Biobank, they screened them for these 32 metabolites.
Okay.
Yeah. So now looking at this picture of the key solute transporters, we can now label them according to whether these have a genome-wide significant hit. In this case, we used a distance of 100 kb. Like, do they have a genome-wide significant hit in around them or not? All of the ones in green do have a genome-wide significant hit, and so there's 8 out of 10 that have a hit. Which is about a 37-fold enrichment. That's obviously enormously significant. And so what you can see actually is that GWAS almost immediately lights up this whole pathway. Like, virtually all of these genes are identified by the GWAS. We can also look at the biosynthesis pathway, and what I'm showing, this is a little bit more complicated. What I'm showing is in red are genes that have hits, and then the gray numbers indicate the numbers of additional genes that are annotated as being involved in catalyzing those steps that do not have hits. And so here we're getting 16 out of 88, which is about a 2-fold enrichment, and that's actually that's a significant enrichment, but it's obviously a pretty modest enrichment relative to what we see for the transport. And so the picture that we get here really is one where the biosynthesis process that produces urate is a relatively weak determinant of urate levels, and actually what really matters is genes are involved in kidney function, especially kidney transport. And I'm going to get to testosterone in a minute, and we'll see actually that for testosterone the picture is very different, but actually biosynthesis of testosterone really is very important. And the last point that I want to make on urate is that although we get these individual big hits that are in these transporter genes, actually most heritability is driven by SNPs and kidney regulatory chromatin. So in this regard, urate looks a lot like other traits where there's a large number of SNPs that are putatively affecting regulatory function. And again, this is really pointing us towards kidney as the key tissue of that's controlling urate levels. So here, this is using stratified LD score regression. We estimate that there's about a 35-fold enrichment of heritability in SNPs that are in active chromatin in kidney relative to a genome-wide background. And then if we remove kidney enhancers from the analysis, then that basically removes the active chromatin enrichment for all the other tissues. So in other words, we can explain most of the signal using kidney regulatory regions, or I should say most of the regulatory signal for urate is active in kidney. I want to give you one more example of a trait before we start to talk about more general models, and that's going to be testosterone. So testosterone is going to illustrate some of the same themes as well as some different themes from urate. So first of all, as you'll know, testosterone has anabolic roles in both sexes, but the overall levels of testosterone expression are quite different. So it's about 10-fold higher in males. So by anabolic, I mean that it's it tends to produce, to cause cellular growth, and in particular muscle growth, and as well as secondary sexual characteristics. Again, we know the metabolic. We know the biosynthetic pathway that leads to testosterone. Again, everywhere there's an arrow on here, we know what the enzymes are that are catalyzing those. If you look at these, you'll see that there's a whole bunch of different molecules that you're familiar with already. So for example, cholesterol here, as well as estrogen-related molecules here, cortisol over here. So a lot of brand name steroid kinds of molecules are in this pathway. So because of the sex differences in testosterone, as well as some preliminary analysis, we decided to look at the testosterone levels separately in males and females. And so this, of course, roughly halves the sample sizes. So we have about 140,000 females in UK Biobank. And looking at this, what you see is that actually many of the top hits are in the ones labeled in green here, contain genes from that biosynthetic pathway. And I'll show you a little bit more about that in a minute. And the other category that contains at least a few of the top hits are genes that seem to be involved in gonad development, as far as we can tell. When we look at the signals for male testosterone, we find that the patterns are completely different. So there's essentially no overlap between the hits that we get in males versus females, and the processes that this highlights in males are generally different. So remember, for females, that this green category that's involved in biosynthesis was the lead category. We do get some hits for males, although they're generally in different genes, and I'll come back to that. Actually, the most important process is a bunch of genes that are involved in regulation of a gene called SHBG, which is steroid hormone binding globulin, and I'll talk about that a little bit more in a minute. That's a protein that actually binds a fraction of the testosterone when it's circulating around in the bloodstream. And so everything on here that has got an asterisk on it is involved in the regulation of SHBG levels, as well as here the actual SHB gene itself.
Jonathan, this is Andy Griffith. I have a question. So the testosterone is free testosterone level?
It is total testosterone in blood. So
okay.
it's including both the free and SHBG bound.
Oh, okay. Thank you.
Yeah. So I've already kind of hinted at this. What we find is that actually male and female testosterone are essentially unrelated traits. And there's another paper out just earlier this year in Nature Genetics that makes the same point. from John Perry's group. So this is a plot where we're taking the hits that are either genome-wide significant males or genome-wide significant females, and we're plotting the effect size of these one against the other, and you can see that the correlation here is essentially 0. And then similarly, if we use LD score regression to look at the genome-wide correlation, we find that again the genome-wide correlation is basically 0. We do not reject the null hypothesis of 0 correlation between the sexes. So this struck us as really surprising, and so we wanted to dig into this a little bit more.
And so the chip heritability is the same per sex?
I'll get to this in a minute. The heritability for females is about 2/3 as large as it is for males.
Okay, so not a big difference. Okay.
Correct. And actually, you can get a sense of that here. So there are 127 genome-wide significant hits for males and 79 for females. And the heritability, sort of the estimated SNP heritability, is roughly proportional to those numbers. Okay, so digging into the data, we think that there's a few different things going on. So the first thing is that steroid synthesis is important for both sexes, but it's actually different parts of that pathway that wind up being involved. So here, going back to that that pathway plot from before, but now we've actually put the gene names onto there, and so they're color coded in the following way. So if there's a hit in males, that's labeled in purple. If there's a hit in females, it's labeled in orange. And then genes with a hit in both sexes are labeled in black. Although the few examples where there are hits in both sexes, like these, it's actually different variants. There's essentially no overlap of variants in this pathway between males and females. Also, if you look at this, you get a sense there's actually different parts of the pathway that are most important. So most of the male hits are down in this lower part of the pathway that are in the sort of androgen synthesis part, whereas the female hits are generally up in this upper part of the pathway that leads to cortisol and aldosterone. And we think that part of what's going on here is that actually there's a difference in the main organs that are involved in testosterone production between males and females. So in males, most of the testosterone is being produced in the testis. In females, a small part is coming from the ovaries, but actually the adrenal gland is much more important, and adrenal is producing aldosterone and cortisol. And so probably genetic variation, this upper part of the pathway, in the adrenal gland winds up having a relatively larger impact on testosterone production for females for that reason. There's also really important differences between the sexes in regulation, as well as I mentioned in tissues of production. So for males, there's this important feedback and negative feedback loop on testosterone production. So as I said, testosterone is produced in the testis, it circulates in the bloodstream. When it's in the bloodstream, about half of the testosterone is bound by SHBG, which effectively makes it bioinactive. And when it's but the active component of the testosterone basically produces this negative feedback loop that's blocking signaling from the hypothalamus and pituitary. And in the forward part of the feedback loop, you get the hypothalamus and pituitary is feeding forward. It produces this hormone called luteinizing hormone that's stimulating production in the testis. Okay, so basically we've got this forward loop from hypothalamus and pituitary that's blocked by testosterone circulating. So if you think about people like Barry Bonds who have taken testosterone for performance enhancement, it's apparently what happens is that that actually leads to shrinkage of the testis, and that's basically because of this negative feedback loop here. In females, the levels of testosterone are so low that there's no real negative feedback. And so even though some of the testosterone is bound by SHBG, that doesn't actually really change the, oh, actually, let me say one more thing. So for males, if you increase SHBG levels, that actually takes testosterone out of the free component, and it reduces this negative feedback loop. And that means you produce more testosterone. So this helps to explain one part of what I was showing you, why SHBG is so important in males, because if you increase SHBG levels, then you have a corresponding effect on increasing testosterone levels. In females, if you increase SHBG levels, it doesn't actually affect the amount of testosterone production because there's not enough testosterone anyway to drive this negative feedback loop. There was these crazy experiments that were done in the 1980s where they actually increased the amount of testosterone in women by about 10-fold. And when they got up to about 10-fold, they were actually able to drive this negative feedback loop. But they had to get all the way up to there for the feedback loop to kick in. So I don't know how they got IRB on that one.
Jonathan, I have a question.
Yeah.
I'm curious about the ranges of the actual values in men versus women. So for example, in men, is the range, you know, orders of magnitude? Is it really high? And in women, are we talking about very small changes? I didn't actually understand the y-axis on that graph.
Oh, okay. So you could think of this just as being like a histogram. So it's a scale density plot. And so, yeah, so basically there's about a 10-fold difference in the mean, and then the distributions in the tails are essentially almost not overlapping.
I see. Yeah, I didn't read that properly before.
Yeah, fair enough.
Sorry.
Okay. Yeah. Thanks for that question. Yeah. Okay.
But I do notice that's a log scale.
Yes, exactly. Yeah. Yeah. So it's about a 10-fold difference in the mean.
Yeah.
So yeah, I think it makes it clear why, you know, when you here, like in males, you know, modest effects in testosterone level having a big difference, but in females, when you're in a completely different part of the x-axis, you know, it's, you know, you're sort of on a different part of that response curve.
Yeah, thank you. That's what
Sure.
I was thinking.
Yeah. So, okay, so that's the picture I just showed you. And then if we look at genes that are in the upper, that are controlling the process in the upper part of this pathway, and so in the hypothalamus and pituitary, we see that in males, a bunch of these genes actually have genome-wide significant hits. All of the ones that are in purple have genome-wide significant hits. Actually, none of these have genome-wide significant hits in females. And that's, again, that's showing that actually this upper part of this forward pathway is also not really doing much to control testosterone levels in females. And we think there's two reasons for that. One is, as I mentioned before, actually the adrenal gland is probably contributing, is believed to contribute most of the testosterone in females. Secondly, the effect of luteinizing hormone on testosterone production, the ovaries are a little bit more complicated, and I don't have time to get into it. But anyway, what we think in summary is that actually the regulatory pathways that are driving testosterone production are, you know, really completely different in males and females, and so they wind up having a, you know, completely different genetic basis. So, so there I've sort of given you a lot of details about the genetic basis of these two traits, but I now want to sort of step back and talk a little bit more about what this tells us about genetic architecture. So first of all, the fact that these are, you know, the fact that they're sort of really lighting up these important biological pathways, does that mean that they're less polygenic than a trait like schizophrenia? And so the quick answer is really not really, or early, yeah. So what I'm showing you here is a plot of the, basically a cumulative plot of the SNP heritability across the genome for testosterone. And so to help you understand what this is, so basically what we've done is we've chopped the genome up into blocks. For each block, we estimate how much that block is contributing. This is like approximately megabase blocks. We estimate how much is that megabase block contributing to the SNP heritability, and And then we're just sort of plotting that cumulatively across the genome. So you can see here that the SNP heritability for females is estimated to be something like 17%. And that's contributed, sort of roughly uniformly across the genome. You see a couple of small jumps, like the CYP3A7, which is marked in orange, and this is one of the genes that is in the biosynthetic pathway. Here in males, you see a jump that's corresponding to SHBG, but overall, the known pathways are actually only contributing about 10% of the SNP heritability for males as well as for females, although the relevant pathways are different. And I won't show it, but we get similar results for urate as well. We've estimated that the number of variants that are affecting each of these traits is somewhere in the 5,000 to 10,000 SNP range. So this is less polygenic than we get for height. So for height, we get about 80,000 SNPs, but it's still implying that there's an enormous number of SNPs across the genome that matter for these traits, and that they're spread pretty, like fairly broadly across the genome, and even these most important pathways are only contributing about 10% of what's going on. Most of what's going on in the heritability of these traits is from outside of the main pathways. So let me recap the first part of this talk, and then I'll tell you a little bit about models of how we're thinking about this. So first of all, what I've argued to you is that for a lot of different traits, we see that the heritability is spread very widely across the genome. I've argued to you that genes with trait-relevant functions and relevant gene sets tend to contribute just a small fraction to total disease risk. Nonetheless, the biggest hits are often highly enriched in most relevant gene sets. Certainly in these molecular traits, we know a lot about them, and there are hints of that in common diseases where we probably know a lot less about how to annotate these. The last point I'd make is that, as we and many other people have noticed, the contributing variants tend to be highly concentrated in active chromatin from relevant tissues. And I think this implies pretty clearly that the immediate downstream effect of most of the SNPs that affect heritability is probably through gene regulation. So any model for thinking about complex traits has to incorporate that. So here's the fundamental question. How should we think about this mapping from genetic variation on the one hand to complex traits, given that so many variants from so much of the genome winds up mattering? So in a pair of papers that we published in 2017, 2019, we sketched out a model that we think can fit some of the observations that we think can fit the observations that we have so far. And so we've referred to this either as the Omnigenic or core gene model. And so here's the idea of this. So I think of this as being kind of a conceptual framework. So in practice, it's sort of simplifying a more complicated reality. But as such, but we still think that as such, it's helpful for providing a framework that we can think about or argue about. So we have this notion of core genes. So a core gene is a gene that we define as having a sort of a direct causal arrow from that gene, either from its protein sequence or its expression level to phenotype. And so, for example, if you think about all of those genes that are catalyzing steps in the female testosterone biosynthesis pathway, those would all naturally be thought of as core genes. And then we think of all of the other genes that are expressed in relevant cell types. So, for example, kidneys for urate levels. We think of those as being peripheral genes. So if you think about a SNP that affects the phenotype, it could do that in one of two ways. It could either be a SNP that's close to a core gene, so it's affecting the regulation or the protein sequence of that core gene. So if you think of the urate transport genes, for example, those SNPs near those can have relatively direct effects. Or if you're a SNP that's sitting close to a gene that's expressed in kidneys, but it's not a core gene, then that SNP might have an effect on urate levels because it's a cis EQTL for its nearby gene, and it's a trans EQTL for genes that are involved in kidney in kidney transport, for example. So, so basically our model is that for peripheral effects, most of these are flowing through gene regulatory networks that are going on inside cells. So we kind of have this, sort of conceptual model where the core genes for a particular trait are embedded in this larger gene regulatory network. And so basically SNPs that are anywhere that are affecting any gene that's plugged into the regulatory network may have non-zero effects on the trait itself. And so this is why we refer to this as the Omnigenic Model. We have this idea that basically any gene that's expressed in the relevant cell type has the opportunity to have a non-zero effect on the trait, regardless of whether it's sort of directly involved in the biology of the phenotype you're looking at or not. And so to try to be a bit more specific, we'd make the following hypotheses. First of all, we think that actually most of the heritability is not coming from core genes, but instead it's flowing indirectly through these weak transregulatory effects. Actually, this is the point I made a moment ago. We think that essentially every gene that's expressed in the relevant tissues has the potential to have non-zero effects. And then because of that, basically any variant, a regulatory variant in a relevant tissue can have non-zero effects. So in general, we don't actually know these cellular regulatory networks in any level of detail. So we can't really evaluate this model directly yet, but I do want to draw your attention to some lines of evidence that bear on this. So first of all, let's think about cis and trans variation that affects a core gene. So core gene by our definition has a direct causal arrow to the phenotype. So then if for SNP variation to affect it, it has to do that either through cis effects that are local or through trans effects that are presumably mediated through expression levels of peripheral genes elsewhere in the genome. So a first question you might ask then is, for a typical gene, how much of the expression variance is coming from cis versus trans effects? And if you've ever done EQTL mapping, you'll know that in general, trans EQTLs have extremely small effect sizes compared to cis. So this is a plot that we made a couple of years ago, but many other people have pointed this out. So in this study, what we did was we identified candidate cis and trans EQTLs in one data set, and then we looked at their effect sizes in a replication data set. And this plot just shows you the cumulative distribution from biggest to smallest of the effect sizes. And so you can see that cis EQTLs have much, much bigger effect sizes than trans EQTLs. And everybody who sees this, who does this kind of thing, finds the same thing. However, what maybe less well known is that actually most of the total variance in gene expression seems to come from trans effects. And there are a number of studies that have found this. So one of the first that I know of is this paper from Alcus Price and others from 2007 or so. And what he showed using Icelandic decode data was that about 37% of the variance in gene expression of the genetic component of variance in gene expression is coming from cis in blood, 24% in adipose, and the rest is coming from the rest of the genome. And so basically the approach there was to look at correlations in gene expression between relatives and then partition that according to whether they're sharing the cis region IBD. So it's actually kind of a very tight design that shows convincingly that actually most of the variance is coming from trans effects. And so I did a survey last year of different studies, both from our lab as well as from others. And although there's some variation across the different studies, all of these are concluding that something like 3/4 of the variance in gene expression is coming from trans effects. So just to put a round number, and let's say about 70%. Okay, so now we can start to put some numbers on the picture that I showed you before. So we might imagine that if a core gene is, if core genes are sort of typical of genes overall, then about 70% of the variance in expression of that core gene may be coming from trans effects. At the same time, we know that trans effects are generally much, much weaker than cis. And so that implies that, putting these two things together, it implies that a typical gene has to have enormous numbers of very, very weak trans regulators. And so if we assume now that there are at least tens and probably hundreds of core genes for a typical trait, and then each core gene is probably affected by perhaps hundreds of transacting variants that are all have tiny effects, now this starts to give us some idea about why so much of the genome may contribute to any given complex trait. And the last point that I want to make on this is to think about how much of the heritability we should expect to come from core genes. And the model, we've done a bunch of modeling on this that's in our paper from last year. I won't go into a lot of detail, it's a little bit complicated. But the key point is that depending on the details of the model, anywhere from about 70 to almost 100% of the heritability for the trait can come from transregulation. And so these different curves basically come to correspond to different parts of the parameter space. And you can see that for a bunch of these models, actually virtually all of the heritability for a downstream trait is coming from weak trans effects that are flowing from peripheral genes. And so we think there's actually probably a lot of what's going on in complex traits. So the last thing that I want to show you is some future directions that we are doing in the lab in collaboration with Alex Marston's lab at UCSF. So I think one of the great challenges right now in this, well, partly in our model, but actually in human genetics in general, is that we know very, very little about these transregulatory networks. And so in principle, you might try to measure these using trans-EQTL mapping, but it turns out we probably need on the order of tens of thousands of individuals to make this work. And this is probably possible for blood, but it's not possible for, for most inaccessible tissues. So an alternative approach is to use experimental perturbations. And so this is what we've been doing. And so there's now a number of methods that are in this space. So you may be familiar with Perturb-seq or crop-seq. So in those kinds of methods, what people are doing is using CRISPR knockouts to inactivate a gene of interest, and then you can do RNA-seq to measure effects on expression. So it's basically taking a gene and looking downstream in the network. We got interested, this is Jake Freimer, who's a postdoc in my lab, got interested in asking the question whether we can actually look upstream in a network. So if you think about a core gene or a putative core gene, how do we identify all of the regulators upstream from it? So the approach that he's taken is basically we put a fluorescent tag on a protein of interest and then put that onto a fax sorting machine. And so then you can pull out cells that have either high expression of the protein or low expression. And then that's coupled with a CRISPR knockout library. And so then you can identify guides that knock out a particular gene that are enriched in the high tail or in the low tail of the distribution. And so basically in the current experiments, what we're doing is making a knockout library against all transcription factors, but we think we can scale this up to go genome-wide in future. At present, it's somewhat low throughput, and since we have to pick a particular target, and then it's one experiment for each target. But we think that what we can do is pick particular targets of interest. We can look upstream, and then we can do Perturb-seq kinds of methods to look downstream. And by doing that, we can actually fill out whole parts of the pathway that we think are of importance for a particular disease. And so I'll just show you a couple of examples of this. This is for the gene IL-2RA, which is an important cell surface immune regulator in T cells. So we've done this with ex vivo primary T cells, and the two axes here are showing you the effects of knockouts in two different donors. And then the red dots are all of the genes that are coming out as significant. And you can see from this that, well, first of all, I want you to see that the data are extremely replicatable across different donors. And then actually many of these genes that are coming out are genes with important immune regulatory function. So actually this makes a lot of sense, and follow-up work that he's done to validate this has shown that most of these validate really nicely. We can also look at this in different cell types. So this is a comparison between the data for T effectors and T regulators, which have somewhat contradictory roles in immune regulation. And we find that some genes have the same effect on IL-2RA in both types of cells, but actually some genes like MED12 have an effect in T effectors, but not in Tregs. I should note that these particular data are a bit more preliminary than the last one. So we think that actually this type of approach in the future is going to be really an important part of the tool set for starting to dissect regulatory networks in a lot of detail. So that there's starting to be a lot of methods in this kind of space, and I think that actually, these perturbation techniques are, going to be as important, if not more important, than trans-EQTL mapping for this kind of work. So, in summary, what I've shown you is first of all, this Omnigenic model, which we think of as it's sort of a conceptual model for trying to develop some framework for how it is that so much of the genome matters for complex traits. So we've proposed that essentially all of the genes that are expressed in disease-relevant cells can affect the regulation of core disease genes. We've argued that probably most of the heritability is coming from SNPs that are outside of core pathways and core genes. We think that most heritability is coming from weak trans-EQTL effects. And then a couple of points I didn't have a chance to talk about are, first of all, we, as well as Luke O'Connor in Boston, have argued that actually there's another piece of this playing out, which is that natural selection probably tends to flatten the impact of the most important genes. And then lastly, I talked in detail about these molecular traits like testosterone and urate, where we think there's on the order of 10,000 SNPs that matter. For complex diseases, if you think of something like cardiovascular disease or diabetes, these are traits where there are actually sort of many different endophenotypes that all kind of feed through to the disease state. So if each of those endophenotypes is affected by 10,000 SNPs acting in different tissues, then you can easily imagine that the end disease state itself is going to sort of accumulate all of the genetic variation that underlies all of those endophenotypes. So that's probably another important process that contributes to polygenicity. And then the last kind of questions are, what's the field going to do going forward? So, one key question for GWAS is to think about how we can infer what are the core genes and master regulators. I mean, I would argue that, finding the gene that's regulated by the 1000th SNP in your GWAS hit probably doesn't really matter if that gene is not really very closely connected to the biology. What we need to be able to do is to look at the GWAS data and pull out what are actually the genes that are the core genes, as well as the TFs that are regulating those, and that's sort of where the biology itself is. I think that these regulatory network methods may wind up being really important for that, although they're still, I would argue, in their infancy. If you think about druggable targets, really you need to know the structure of the network and the core genes to really think about how you would drug that system. And then the last point I would make is that here I've sort of been talking throughout about how do we get biological insight out of disease GWAS, but of course there's another use case for GWAS, which is polygenic scoring for prediction. And for that, actually, we really don't care about mechanism. We really just care about having a very large sample size so that we can estimate SNP effect sizes as well as possible. So, like a SNP with an effect size of beta on a peripheral gene is as important for the polygenic score as a SNP with an effect size of beta on a core gene. And so, polygenic scoring, there are a lot of issues, but I think that, what I'm talking about is less relevant for that. So I'm going to end here, but I would like to acknowledge the people in my lab who've driven a lot of this. So as I mentioned, Evan and Yang, who used to be in my lab, Evan's now a postdoc at the Salk, and Yang's an assistant professor at the University of Chicago. They really drove the beginning of this project. Chuan Yao and Yang led the second paper with a lot of the theoretical modeling that I described. Troy Niao is now also an assistant professor at University of Chicago. NASA and Sahin are in my lab currently. They've done all of the work on the molecular biomarkers. There's a bioarchive preprint if you're interested in it. In that, and it's got quite a lot more detail than what I was able to tell you. And then Jake, who's in Alex Marston's lab, has done all the CRISPR perturbation work. And here's a picture of our lab Zoom from a couple of months ago. So I'll end there, and hopefully there's a little bit of time for questions. Thanks so much for your attention.
Thank you very much, Jonathan. That was excellent. We'll have 5 or 10 minutes of questions with everyone, and then we'll switch over to sort of the smaller groups. So any questions? I can see Rob has a lot in the chat.
Okay.
Yeah, it might. So my question, basically, humans are a damn weird species. We know it. We're proud of it. So the question is an omnigenic model good for other species that have not been through serial bottlenecks, followed by relaxed selection, followed by ridiculous GXE, because we're panmictic, we're all over the world. We have thousands of different occupations, diets. So I'm just wondering to what extent this generalizes as a biological principle or a fluke of humanity.
That's a great question. Actually, let me go to. So I would say two things are slightly contradictory. So first of all, I think that this principle that trans-regulatory variation is important, or at least we think it's important. That's something I think should be generally true across species. And when people have looked at the contribution to gene expression heritability through trans effects in different species in mice and in yeast, I think are the main of the two non-human examples I know of. You know, they've gotten quite similar numbers there. So I think that kind of aspect is likely to be shared across species. However, the relative importance of core genes, that may depend to a significant extent on the nature of the population history and the strength of selection and the types of selection. And you know, if you compare, for example, to like body size in dogs, right? So body size in humans has been or you know, BMI, for example, like that's more polygenic than height. There's probably well over 100,000 variants. In dogs, there's you know, something like Elaine Ostrander's work has shown something like 5 or 6 variants on their own are enough to explain about 80% of the variation in body size in dogs. Like IGF-2 on its own, I think, explains about 50%. So, you know, that's saying that I think it's saying that if you have a breeding system, of course, in that case, it's like an artificial breeding system where you're taking these freak variants and driving them to high frequencies in you know, in these founder populations, and you can create a different kind of architecture. I mean, my guess is that probably there probably are weak genome-wide effects for body size in dogs, but they're only contributing you know, maybe there's 100,000 weak effects on body size in dogs, but they're only contributing 10% of their variance because the biggest effects are so big. That would be my guess.
Thanks. Perfect.
I wonder if that's telling you something about the core genes. So those sort of ones that are being in dogs or in sort of inbred mice, because they're having a big effect. And the reason you're not seeing it in the human population is because those have been dropped down by selection.
Yeah. So I think there's a couple of different kinds of things that can go on. So one is that like it's pretty clear now that there's like for a lot of traits there's stabilizing selection, so that you have basically like you've got sort of selective pressure towards some optimum, and the net effect this has is that it tends to dampen down the allele frequencies of the biggest variants, and so in some parts of the parameter space you can actually, like, so for the variants with the biggest effects, you can actually wind up with an inverse relationship between squared effect size and heterozygosity, which means that the contribution they make to heritability is just sort of flat. And so basically, like, that kind of purifying selection prevents core genes from sort of contributing as much as you think that they might. So if you can weaken selection, you can potentially increase the effects of those. The second kind of thing is, of course, if you have positive selection in favor of particular variants, then that can play out differently in different ways. So if you have sort of natural selection acting on one trait, then it should sort of shift the allele frequencies of everything kind of proportionally to effect size or squared effect size. But if there's pleiotropic effects, that can sort of distort the relative amounts of movement of different kinds of variants. And then I think in dogs, actually, you have probably a lot of what's going on is it's artificial selection, where you might actually just sort of have somebody who recognizes a really unusual mutation, and then they breed from that and preserve the mutation. That's a process that I don't think it's really analogous to something that you get in the wild. But I think it's probably really important for the architecture in dogs.
Thank you.
Yeah.
Other questions?
Sorry.
Go ahead.
You have shown in one of the slides like about upregulation of testosterone levels in males through HBG axis. So, and also you have shown in that like LEPR, is that your leptin receptor pathway? What you showed is different between two sex?
I'm sorry, I didn't catch all of that. Could you repeat that, please?
Yeah, so you have shown one of the slide where you shows upregulation of testosterone levels in males, right? And then you show LEPR. Is that your leptin receptor pathway that you have shown?
I don't. So I'm just
highlighted blue one in males. You see right side LEPR.
Yes, it is leptin receptor.
Okay, so my question is if we compare metabolically active tissues like skeletal muscle, liver, and adipose tissue, you will see sometimes within the same genotype different phenotype between males and females. So my question is it, I know it is a broad question, but just I was wondering if you have like kind of idea if it is just because of testosterone level upregulation, or it could be due to progesterone versus estrogen. Because one of the very important thing that I have seen in your slide is leptin receptor pathway is different, and that is another thing in metabolism that is very important. It makes sense once you have two different phenotype in male and female.
Yeah, that is an excellent question. And so NASA, who's the student who's done a lot of this, has done some work on using Mendelian randomization for male and female testosterone to try to test effects of this on different phenotypes that have sex differences. But I'm sorry, I can't remember details on that because none of that went into the paper. It's an excellent question. Thank you for that. If you have a lot of interest, you can email me. I can.
Oh yeah, sure. I will email you.
Yeah. Thank you for that.
Thank you.
Dr. Pritchard, I have a question. You mentioned that getting eQTLs can be somewhat difficult and hard to access tissues, like the brain. I was wondering how much does the heterogeneity within specific tissues affect these QTLs, and it is important to distinguish between cell types and maybe organs that have a lot of cell types. So like the brain is like the obvious one I can think of, maybe like the pancreas or something. But I'm just wondering, is like single cell important in this, or does it not matter really?
No, I think that's a great point. And yeah, so if you think of something like GTEx just worked with bulk tissues, and you know, that's just because of where the technology was when they did that. That, you know, people are now just starting to move into using single cell sequencing for EQTL studies, and you know, I mean, that's definitely an important kind of shift. You know, I think that'll provide a lot more power for identifying variants that have effects specifically in minority cell types. So anyway, so that's clearly a weakness of the current studies. That said, I think that it's not clear that that's going to do much to improve like power for trans EQTLs, because I think that those effect sizes are just sort of fundamentally small effects, and you know, what we would really need probably is, you know, is very large sample sizes to solve that problem. You know, very large sample sizes ideally with single cell sequencing, but you know, for a lot of tissues, I don't really see how we can get to that.
Yeah, I mean, you were talking about the testosterone hormone earlier, and you know, steroid receptors can affect transcription totally differently in different cell types. So I was just, that's just why it popped into my head. But
yeah. No, it's a great point. And actually there's another conundrum which we're also thinking about, right? Paradox, which we're also thinking about right now. I don't have time to talk about this, which is people generally find that there's, although we think that most non-coding variants that are GWAS hits must be acting through gene regulation, the overlap between GWAS hits and EQTL is actually surprisingly pretty low. Mm-hmm. And so one part of the explanation for that is presumably what you're, you know, the point you're alluding to, that, you know, when you look in bulk tissues, you're like you're missing a bunch of EQTLs that are only active in very specific cell types that that might be rare in a bulk tissue. So
okay, yeah, thank you.
Sure.
Okay, so if we call that the end of the main seminar and let most of you run away, anyone else who wants to stay and ask Jonathan more questions, and all the students, please stay. We've got Jonathan for another 35 minutes, and I would like to take full advantage of that.